Evolutionary biologists have spent generations studying the changes that developed in living organisms and tracing them back through the history of the Earth. These new developments have been known to be caused by circumstances such as natural selection, genetic complexities, mutations, and genetic drift. Membraceti, or rather treehoppers, carry a viable example of developed changes in the form of their abnormal morphological structures known as their helmets, which can be witnessed to be present here on the dorsal region of the treehopper. Research scientists have been studying this form of helmet morphology and how it has evolved within this species. This can be seen evident within Figure 2 that clearly illustrates the T-segment arrangements. It has been considered that the helmets be merely an enlarged dorsal face of the first thoracic or T1 segment. As the helmet contains the presence of a jointed articulation, it can be deemed to be only a simple outgrowth with a bilateral origin, rather than it being classified as an appendage of the T1. The only other found dorsal thoracic appendage in insects has been wings, which can be found on the T2 and T3 segments. This leads scientists to consider the likeliness that the insect's helmet may have evolved as a set of pervaded wings. This is further supported due to the research that finds that the helmets and wings both share several unique morphological features. As you can see in Figure 3, both share similarities in their hinge regions. Also, they both contain two layers of epithelial cells that are found to be interconnected by cuticular columns. It has also been observed that both features are covered by a complex vein network system. To further prove that the wings and the helmet are developed from the similar origins and that they are in fact serial homologs, they would be required to share genetic programs throughout their development. As you can see in Figure 4, by using the transcription factor of nubbin, scientists are able to observe and distinguish between wing development and other appendage development. By using cross-reactive antibodies to nubbin, it can be witnessed that the developing helmets contain traces of nubbin and that its expression parallels that of the developing wings. This is evidence that represents that both wings and helmets throughout their development share the same genetic program, further supporting the notion that the helmet is in fact a T1 wing serial homolog. The research topic of this article has been used and cited within a number of 22 other research articles. The majority of these articles have used this paper as a reference only, although one article was written as what can only be interpreted as an updated version of the original article. This updated article provides evidence to suggest the need for improvement in the data of the original article, as it implies conflicting results to the original.